So this section is going to be about the flow of information. The information is going to start in the DNA, in the genes, and it's going to be expressed then in the rest of the cell or in the rest of the body. So genes control phenotypic traits. Through the expression of proteins. So if you've got dark hair, you have more melanin in your hair, and your genes control the amount of that dark protein that you make. And there are two processes we want to talk about here. We want to talk about transcription, which is the synthesis of RNA under the direction of DNA. So we're going to use DNA as our template and make RNA. And RNA is made by RNA polymerases. When we synthesized DNA, we used a DNA polymerase. So to make RNA, it's an RNA polymerase. The other word we want to look at here is translation. Translation is the synthesis of proteins. And we have the definition here of a gene. A gene is going to be a region of DNA that can be expressed to produce a functional product. And it may be either a polypeptide, which is a protein, or an RNA molecule. And there is a hypothesis that for each gene, we'll have one polypeptide. So for an individual gene, we get a specific polypeptide. And we have lots of different words for seemingly the same thing. A polypeptide is a chain of amino acids, and it's a protein. Now we have a little diagram, but we could talk about where these things happen. So the top there is DNA. And the DNA is going to say safe in the nucleus. We're not going to let our DNA roam wild through the cell where it might get damaged. Instead, we take the DNA and we transcribe it. Into RNA. And then we send the RNA out into the cytoplasm. Where the RNA will be translated into protein. So let's look at the words transcription and translation here. Transcription, if you were to write down the words of my lecture, you would be transcribing my lecture, and you would be writing down the same words that I say. But if you translated my lecture, maybe you're going to translate my lecture into French. You would be changing my lecture and changing it into another language. Now, the genetic information is written in codons, and it's the sequence of DNA, of the nucleotides in the DNA, that's going to give us the code for constructing our protein. So, translation requires the conversion of nucleic acid language to polypeptide language. And so there's a change in language in translation. Experiments have shown that the flow of information from gene to protein is based on a three nucleotide code. So we call it a triplet. So the genetic instructions are written in DNA and then in RNA as non-overlapping 3-base words. We call these words codons. The genetic code it dictates how the codons are translated into the amino acids. So the genetic code is a set of rules that dictates the amino acid translations for each of the mRNA nucleotide triplets. So nearly all organisms use an identical code. And this is the code. It's right down here. This is like the Rosetta Stone from nucleic acids to proteins. So if you have a code, let's say we've got I guess a G, C, doesn't really matter, U. This is RNA, so we've got uracil. Okay. We start here in the middle, and we hit the G, and then the next letter was C, and the last letter I picked was a U, and so we're going to make this will code for alanine. You can see there are lots of duplicates. I could have had G, C, C, and I've also get alanine. If I picked code that was, for instance, let me change colors here so we can. How about pink? That'll be different enough. Let's say we picked U, A, A. So our code was U, A, A. That codes for a stop. And that tells the enzymes that the protein is finished. So many amino acids are specified by more than one codon. And the stop codon marks the end of the gene and causes transcription to stop. So here we have another diagram, except we now can see the base pairs. So this is our DNA. It will come apart, and this RNA will be made. So the T here will code for that A, and this A will code for this U. This C, in the DNA, it codes with G, and over here in the RNA, it will code for the G. So we've made RNA, and then if we look on our Rosetta Stone, AUG spells methionine, AAG spells lysine, and UUU will spell phenylalanine. Transcription produces a genetic message in the form of RNA. In the nucleus, the DNA helix unzips and RNA nucleotides line up, and the RNA polymerase joins them in one strand along the DNA, following the base pairing rules. A specific nucleotide sequence called a promoter acts as a binding site for the RNA polymerase and determines where transcription starts. The RNA polymerase adds nucleotides until it reaches the sequence of DNAs called the terminator. So think Arnold Schwarzenegger. Eukaryotic RNA is processed before leaving the nucleus. So once it's been made, this RNA, right here, before it gets into making a protein, it has to be processed. And what we'll find is that it's all gets spliced up. Introns are non-coding sections, and they are removed. And the exons, the parts of the gene that are expressed, are spliced together. We also add a cap and a tail. So let's look at the image of that. We've got a cartoon. So here's our DNA, the blue. We have transcription, and we get the cap added, and the tail is added during the transcription. And then the introns are removed, the light pink parts, and the exons are spliced together. And once it's been processed, the mRNA leaves the nucleus. And we call this messenger RNA. We're going to have 3 different types of RNA. And that's because it conveys the genetic message from the DNA to the ribosome and the translational machinery of the cell. The second type of RNA we're going to have is tRNA, and this is transfer RNA. The transfer RNA is going to serve as an interpreter during translation, and this takes place in the cytoplasm. So the ribosome is going to attach to the mRNA, and it's going to translate the message into a particular peptide, and this is going to be aided by tRNA, or transfer RNA. tRNA is going to function by linking a particular codon on the ribosome, on the mRNA, to a particular amino acid. And we have here 3 different representations of tRNA. All 3 of these are tRNA. The first just shows its sequence. It ends up kind of in 3 big loops. The most important parts are this anticodon. This is the part that's going to match up with the RNA, the messenger RNA, okay? The other important part is this amino acid attachment point. This will have only one type per tRNA amino acid, and it will match the anticodon, so that this is what actually does the translation. This is the second one here is a more folded vision of the same molecule. And lastly, this is the one we'll use. It's kind of a cartoon version. You have the anticodon down here, and we've got an amino acid site, so an amino acid will be attached right here at the top.